Welcome everyone. As everyone gathers, we will wait for a couple of seconds just to get going for this monthly seminar series. Just like most times, if you want to ask a question at the end from Dr. Rehm, you can either put your question in the chat and I can ask it, or we will open up. If you're brave enough, you're welcome to ask a question directly. So it is 4:01, so we will get going with our speaker this month, who's going to be Dr. Heidi Rehm. She is the Chief Genomics Officer in the Department of Medicine and the Center for Genomic Medicine at Massachusetts General Hospital, where she works to integrate genomics into medical practice, which she will be telling us about today. She's a board-certified laboratory geneticist and medical director of the Broad Institute Clinical Research Sequencing Platform, working to guide genomic testing for clinical and clinical research use. She's also co-director of the Program in Medical and Population Genetics at the Broad Institute and professor of pathology at Harvard Medical School. She is a principal investigator of ClinGen, which I think many of you on the call know, providing free and public accessible resources to support interpretations of genes and variants. Dr. Rehm also co-leads the Broad Center for Mendelian Genomics and focused on discovering novel rare disease genes, and co-leads the Matchmaker Exchange to aid in gene discovery. She is a strong advocate and pioneer of open science and data sharing, which I think we all appreciate, and is working to extend these approaches through her role as vice chair of the Global Alliance for Genomics and Health. And Dr. Rehm is also a principal investigator of the Broad LMM Color All of Us Genome Center, supporting the sequencing and return of results to a cohort of 1 million individuals in the US. She also co-leads NOMAD, which Nomad, I was reading my notes. She co-leads Nomad, which I'm sure many of you have used. Anyone that's doing genetics obviously would have used that in the genome aggregation database. As you can see, she is incredibly busy. So we are super happy that Dr. Rehm accepted the opportunity to speak to us today. I should say this led out of a webinar I moderated about 2 months ago, where Dr. Rehm was one of the speakers. It was a panel on population genomics, but at the end, it kind of, Heidi moved it towards clinical utility, clinical interpretation. And I said to her, wow, it'd be great if you could come back and speak on that directly, because I think it's something that a lot of places are grappling with. And so Heidi, thanks very much for accepting the opportunity, and go ahead.
Great. Well, it's a real pleasure. I appreciate the invitation and an opportunity to speak with you all. It's funny you brought up that impetus for this talk, because I went in to put these slides together for this talk, and I always cut and paste the email conversation that led to an invitation into the meeting hold for the talk slot. So I opened up like, what did I say I was going to speak about? And there was this email from Adrian, and it actually said, I think many, Pitt would like to hear your vision as you quickly stated at the end. And I thought, what was I talking about? And I couldn't actually, I still can't totally remember what I was talking about, but you went on to say how you've set up research in clinical genomic sequencing. So I made a little bit of a guess as to what I was talking about, and we'll actually focus more on really what we're doing at the institution and my role as Chief Genomics Officer at Mass General Hospital, because I think that touched on, but what I was sort of talking about at the end. But I have left enough time. Hopefully, I've timed it for like at least 15 minutes worth of discussion. So if I miss the boat and you guys want to ask more questions, we should have time to discuss. So with that, I'm going to just continue on. So as Adrian mentioned, I work in a few different spaces here, both in discovery and mostly rare disease on the discovery side, but I also work, in various genomic resource building databases like our own biobank, eMERGE, all of us. Also work a lot in implementation. And I'm going to talk in particular about this stuff at the top, the Genomic Medicine Unit at Mass General Hospital, touch a little bit on the Broad Institute and our sequencing platform. For rarity, not talk much at all about ClinGen. I think I have one slide in there about ClinGen. There have been some other studies I've done collaboratively on implementation. And also, as was mentioned, I do spend a lot of time in building standards and data sharing platforms, because that's really critical for all the work we do in this space, is data sharing. But I think in terms of the perspective I'm going to share today, a little bit of history as to where I got to the sort of perspectives that I have right now in thinking about how to support genomic medicine in, in the healthcare setting. So I've been at Mass General Brigham, which is the current new name for Partners Healthcare, which is an umbrella organization with many hospitals, the two primary ones being Mass General Hospital and Brigham and Women's Hospital, but there's about 18 other smaller hospitals as well. And I've been in that system for 23 years now. And my first faculty role was back in 2001 when I was hired by Raju Puchalapati to be part of the Harvard Partners Center for Genetics and Genomics. And that was a whole effort to really invest in human genetics at what is now Mass General Brigham. And they set aside $80 million to do this. And that included building a lot of infrastructure for genomics. It included developing core facilities in genotyping, sequencing, proteomics, other omics, demonstration projects, a lot of information, IT technology and infrastructure and bioinformatics pipelines, building a biospecimen repository that is eventually led into the Mass General Brigham Biobank, a lot of educational opportunities, and a part of that was building a new clinical lab. And that's what I was hired to build, was the Laboratory for Molecular Medicine. And the goal of that lab was to translate novel discoveries into cutting edge tests and accelerate the adoption of molecular tests into clinical care. We intended to address areas with little or no available diagnostic tests, improve existing tests with cutting edge technology, support collaborative opportunities with investigators at our institutions, and then support the adoption of evidence-based genetics and personalized medicine. So that was the intent. And we launched this lab. I was hired at the beginning of 2002. It took a little over a year to really get all the regulatory approvals and launch this lab, which happened at the end of 2003. And then we started launching tests, and we did it very rapidly. We launched the first test for lung cancer treatment within 3 months of the publications of EGFR's role in lung cancer. We had a test worked on jointly with the two pathology departments and the LMM to launch this sequencing-based test. We launched the first clinical test for hypertrophic cardiomyopathy back then. And back then we were still doing Sanger sequencing. So launching a test sequencing 20,000 base pairs per patient was actually a pretty significant technology feat. Now that lab, as I mentioned, launched in 2003, and we rapidly grew over the next 5 years. We went from employee 1, which was me, to probably about 50 at the peak, 50 employees, not counting the sequencing core that was doing the sequencing for our tests and other, the biosample repository that helped with intake of samples. So, there's probably more like 100 employees that was touching in some way, shape, or form this clinical effort. But then after about 5 years, testing began to plateau in terms of the clinical side. And over the next ensuing, 10 years, gradually became replaced by mostly NIH-funded research projects like MedSeq, BabySeq, eMERGE, now all of us. And the reason for that was the commercial reference laboratory business was growing, and these companies began managing insurance billing in particular, and they were eventually capping patient out-of-pocket expenses. And this was, and still is to this day, enormously attractive to patients and physicians. It is a morass to figure out whether you're going to get a bill after genetic testing when you go into a hospital. And the physicians can't help you figure out whether you're going to get a bill or not, and how much that bill may be. And it may be even way more than the laboratory actually charged because of how CPT code rates are negotiated between hospitals and insurance payers. I've had at least 3 cases of patients getting $25,000 bills for one test that my lab actually only charged $2,000 for. So this was a huge issue. And as the physicians realized that these commercial labs would cap and say, no matter what happens, you won't pay more than X $100. And today it's about $250 for most panel-based tests that they cap that out-of-pocket expense. That essentially, over time, fairly quickly wiped out all of our clinical business because of that billing issue, which we were not legally able to cap a patient's bills. And then, more and more labs got on the market. Competition has continued and has really driven prices to below cost. And I would probably argue that most, if not all, the currently operating labs operate at a loss, including the largest ones. So eventually I got frustrated with this. And in 2018, I stepped down as director of the LMM. I moved on to a role as a chief genomics officer at Mass General Hospital. Why did I do that? We built a clinical lab that could do any testing in clinical genomics, and not just this billing issue, but I really still saw that patients still had limited access to clinical genomic services. And I wanted to shift my efforts really to the front lines to address barriers in patients and physicians being able to consume genomic medicine. And that's been some of my focus on the clinical side since I took that role. And part of that role was building the genomic medicine unit at MGH. And the goal is really to better integrate genomics into the practice of medicine and through engagement with the key stakeholders, identifying pain points, and delivering new solutions. So I'm going to talk a little bit about some of the work that we've done over the last couple of years. On the left is sort of my core team of both genetic counselors, genetic counseling assistants, couple of physicians that are particularly part of our preventive genomics clinic that I'll talk about, and clinical laboratory geneticist Chrissy and myself. But then with Katrina Armstrong, who's the chair of medicine at Mass General Hospital, with her support, we really reached out to all the divisions in the Department of Medicine. And then I continued onward to all the departments across the hospital that have any engagement with genetics, and identified physician leads for every division or department. And in places where they had genetic counselors, identified a genetic counselor lead as well. And we formed what we call this genomic medicine implementation team that represents all of these different specialties. And initially we started gathering monthly as we were getting things off the ground. Now it's a quarterly meeting with this whole group. And as we, in the beginning, I met with every one of these leads that the chair of the division or department identified, and really got a kind of a landscape view of what was happening in their division or department, and what were their pain points. And that, they identified referral and triage challenges, knowing which clinics to refer a patient to, particularly in primary care. Where do I send this patient who just told me they have a risk of a family history of cancer? To the cancer clinic, to somewhere else? Like, where do the patients go? Lack of genetic support for clinics with low volume genetic testing needs, where they can't really justify or hire a full-time genetic counselor, but they still have needs. So where do they get that support? Certainly appreciation that genetics has been underutilized in primary care, and we'll get into that in particular. And to that point, there's a high volume of asymptomatic patients with genetic risk, but no clinic specifically addressing their needs. Also, the inability to actually understand genetic testing utilization across our hospital. I'll talk in depth about that. Many reference labs in use, send outs and results not being tracked. Inconsistency of obtaining consent for testing, and lack of support for ordering and prior auth. All issues. So we tackled a number of these areas. The first one we started was just a Mass General Brigham-wide consent form, and we formed a core team. We developed the consent form, tried to get it to 2 pages, which it is front and back, and then went through the various reviews and approval process throughout the hospital systems. That took about a year, and it's now approved and used widely. It's translated into 6 languages, and there's policies for use around it in the hospital. We also, in the process, removed the break the glass policy, which was currently protecting genetic results in the hospital Epic system, not allowing access to anybody but the ordering physician. And we argued, look, if we're actually going to find utility in genomic medicine, the entire healthcare team needs access to these results. And so we removed break the glass that was put into place a number of years ago. I mentioned this low volume genetic service needs for genetic counseling. And so we created a model for floating genetic counseling services where I have a team of genetic counselors that can be deployed for clinics that just want on-demand genetic counseling. I'm going to see a patient in 2 weeks. I'm going to need to order genetic testing. Could someone come and help me out, and so on. So we did that. The first real rigorous one was with the renal division, and actually Andrew Lundquist, who we worked closely with, wrote a paper about the success of this model, and allowing them to really launch utilization of genetics in their clinic. And now all of the other renal nephrologists are now using genetics as well. We also, for other clinics that have slightly higher demand than this floating need, have been assisting in hiring and splitting efforts. So maybe a clinic needs a half an FTE, and we divide that between 2 clinics and sort of manage the distribution of GCs. So that's how we've been managing some of those services. In addition, I mentioned lack of awareness of what clinics are out there and where to send patients with genetic questions. So we surveyed all of the clinics and who was seeing, had some specialization in genetics, and then created a web page for Mass General Hospital on all of the genetic specialty clinics. There's 11 of them, representing 35 physicians who see patients, 38 genetic counselors, and over 25,000 genetic tests ordered per year. There. And we created this website. We have a single central contact point for questions, although you can also just click on the clinic and go directly to their website. But patients who aren't sure can call us, and my team helps triage which clinic they might go is most appropriate. I mentioned that the care of asymptomatic but at-risk patients was a huge issue in terms of where to send these patients. So in the end of 2019, we launched a preventive genomics clinic, really to address this growing need, this patient population. This is the clinic you can see on the right when we opened it, and the team that is supporting it, which is essentially the core team I showed you in the beginning, are supporting this clinic. The goal of this clinic wasn't to, as I sort of alluded to, wasn't to address the healthy, wealthy population who wants their genome sequenced, although we do support that. But it was more to address this, first topic here. Someone who comes in and says, my sister died of breast cancer, should I get a genetic test? And says that to their primary care physician. What's the answer? Should they not? What test? And the primary care physicians are not always adept to answer those questions. But yet our hereditary cancer clinic is so overwhelmed that they cannot see all of these patients who have a risk concern, but haven't developed cancer. And most of them, they are actually not able to see. We have like 200 to 300 patients a month with this as an indication, and we're desperately trying to figure out how to support them. Also, patients who have existing genetic test results but need guidance, or as I mentioned, the sort of curious, concerned group. So what have we seen so far? About half of the referrals to this clinic come from primary care, which is exactly what we expected and hoped for. About a quarter are self-referrals. They've found us and called us and wanted to see us because of their own concern, or about a quarter from other subspecialists. And that might be that a patient was seen in cardiology, but then mentioned that they also have a risk for cancer, and can they also get a genetic testing for cancer? And the cardiologist is like, not my job. And so they might refer a patient on. We've seen about 153 patients. That's been mostly during the pandemic. So we had a little bit of a shutdown period and converted the clinic entirely to a telemedicine clinic. There's some patients seen in person, but this actually, preventive genomics lends itself well to telemedicine, it turns out. No surprise there. Genetic testing has been ordered for about 77% of the patients, 13% with multiple tests being ordered, and about 80% of the patients have paid nothing for their genetic testing. And that speaks to the population we're largely serving, who really are at risk and have valid reasons for getting genetic testing. We're still working on diversity. There is a high percentage of white individuals, and this is an adult clinic right now. So the referrals broken down in the areas I mentioned we're supporting, about half are based on family history, a quarter based on some personal history. And because we have a cardiologist who is one of Amit Kara, has been seeing patients, some of the patients do have their own cardiac symptoms that have been seen here. And then about 19%, the curious concern, want their sort of genome sequence or a ACMG panel sort of thing. And then about 7% have come with prior genetic test results, either from a research study or because a family member or somebody else has stimulated them to get testing and it was positive and now they need to talk to someone about it. Now, that's the clinic. We also realized that ideally we would also support education of primary care so that they didn't have to refer everyone to us. And in fact, our clinic is small. It can't accommodate 200 to 300 cancer patients a month. So really in working with particularly primary care to support the utilization of genetics in primary care has been also a goal of ours. And so we launched an e-consult service in Preventive Genomics. This is a screenshot within Epic of the order for the consult service. And they can pick high-level reasons for e-consults, utility of genetic testing for a specific disease or family history, et cetera. So we launched that. We realized that there was some confusion because about the same time the medical genetics clinic launched their e-consult service, and then the cancer clinic launched, a hereditary cancer clinic launched one, and the PCPs were confused as to which service to use. So we actually worked together, all three clinics, combine them into one genetics e-consult, and then my team manages the triage. So we look at the consult that comes in and then decide which service to assign it to, and that's been quite effective. So across the e-consults, we've done 79 to date. Similar breakdown to what we see in the preventive care clinic. The majority are for actual family history, 13% for prior genetic test results, 29% for personal history, and then 2% just coming in for genetic testing without any risk defined. And then we also, because of Amit Kara's particular interest in work in this space in polygenic risk scores, have piloted a utilization of PRS in this clinic. And so we offered, or Amit offered patients access to Color's combined monogenic cancer, coronary disease risk assessment that looks at both monogenic cause of disease as well as polygenic causes and gives a consolidated report. That was launched at the end of last year, and about 56% of patients have had a change in management based on that. I don't think any have come back positive for monogenic causes. They're all based on polygenic scores, and 92% of patients surveyed felt they learned valuable information. And, I think the jury's still out on the true utility of polygenic risk scores, but there is some data out there that really does support their use, but lots more data needs to be collected. Now, I periodically go back to the implementation team I mentioned that represents all the different divisions and departments and ask them to rank their most prominent pain points. So in the most recent survey that I did, really epic integration of genetic test results was the top priority, as well as support for billing and insurance inquiries, and then sort of going down the line there, GC and GCA support, provider patient education, and data collection for clinical and outcomes research. So we are really trying to tackle this top ones, and that led us to this project, which I've been calling the Germline Genetic Testing Consolidation Project. And here, most genetic testing in our hospital system has been sent to commercial reference labs without any tracking by the hospital. And there's multiple factors for that. The complexity of the lab specific requisitions and consent and billing forms, and sample, collection issues. Some of these labs sending at-home collections for both saliva as well as sending people to draw blood from their houses. Our pathology department was just like, we can't manage all of this. You're gonna have to do it on your own. Or the other primary reason to not go through the send out lab in the path department is actually avoidance of any chance of that patient getting billed for testing. And I mentioned earlier that an unexpected bill for $25,000 or some large amount is really just unacceptable, as you can imagine. And so all the physician has to do is get burned once, and they're like, I am out of here. So all of these factors have led there to be a complete wraparound of ordering genetic testing directly from the labs getting PDF reports, and then just they go and upload them to the media tab. And that's how genetic testing has been largely managed at our institution. This, however, makes it very difficult to understand utilization and improve the process if it's entirely outside our system. We did try to do a little bit of assistance, launching a REDCap database to collect basic information about genetic testing across the clinics. It's actually based on a database that our cardiology division launched because they wanted to track this information themselves and had no other way to track it. And then we took a version of that, made it more generic, and supported launching it across multiple clinics. It allows easy query for getting statistics of genetic testing. We can customize it for each clinic and their indications and their commonly ordered tests. And the data entry is about 5 to 10 minutes per patient. And we've actually been supporting some of the clinics and just that we support and entering the data for them. That said, it's not comprehensive. It does not capture, a lot of the test ordering across the hospital. So it's not a permanent long-term solution here. So, moving on to the primary goal here is around better integration for genetic test ordering and results. So we launched this project to identify and consolidate sendouts to a small number of preferred labs to drive efficiency and quality and motivate adoption of our requests of the labs, like EHR integrations, electronic orders, getting structured results back, access to raw data, all of those things that we want as a system. But doing this across 100 different labs is just infeasible. And so the goal was to really look at all the labs that we were ordering tests from and bin them into preferred, acceptable, and discouraged. And the end goal was this, is not to force physicians to have to order from a particular lab, but instead educate them on these labs and their pluses and minuses, and then incentivize them to order from the preferred labs by creating ease of ordering and getting access to results, et cetera. But to figure out which labs, we had to do a lot of data gathering. And we formed a committee under the hospital leadership and got assigned a team to gather data. This is the team. And so we basically made a list of all the major clinical labs we could think of, or we were aware from our colleagues that were in this implementation team where they're ordering tests. And then this small project management team assigned to this went out and asked the labs for our own volume. That was the only way we could get the data, was to go to the labs directly and ask them how much we were ordering from them. And so here's a slide with the 15 top labs that we collected data from over about a 3-year period. And we gave them CPT codes particularly for the labs that do more than just genetics to define which tests. We actually excluded cytogenetics and prenatal NIPT tests, but all of the others were fair game. We also collected some data on quality of lab, which is admittedly a bit subjective from experience of clinicians and et cetera, whether they share data in ClinVar. As those of you who know me know that's a pretty important aspect. Do they have an interface with Epic? The only one we currently have has been with Mayo, because there are contracted reference lab for pathology, like broad pathology services that are sent out. And so that was the only integration. And then the rest of them were really around support for billing and prior auth activities. So those were factors. So this now comes down to the data, and I've binned these into 3 buckets. Those labs that had the broadest test usage. And so when we got the results back, we binned them into these major clinical areas, the team did, so we could track across different disciplines. And so you can see for both Invitae and GeneDx, they had testing in every area. Most of you know that. We then had moderate volume or specialty labs. So this is an example of 5 different labs that had particularly high volumes in certain areas, but weren't very broadly utilized. Ambry has been one of the major labs for our hereditary cancer center, and that's most of the volume from that lab. Prenatal for Myriad Women's Health, which bought Counsel, that's the old Counsel, and Natera and Cooper around pre-implantation genetic diagnosis. Mayo's volume is largely very small, like genotyping tests, factor V Leiden, hemochromatosis, other sorts of tests that are small that fit into that sort of pathology send out bucket. And then we had a number of vendors that we considered low volume tests that just didn't have more than a few hundred tests per year. So that's how we saw the data. We then assembled the data by specialty and picked cardiology, prenatal, hereditary cancer, ophthalmology and hearing loss, pulmonary, neuro, and medical genetics. And we had meetings with teams from those divisions. We shared their specific test ordering practices with them. And then we went through and queried them for the most important factors. Why were they ordering their testing from this lab or that lab? And got feedback around, and not surprising to the early issues we gathered, but the lab's billing practices and transparency, supporting that, whether they took Medicare or Medicaid, whether a lab will begin benefits investigation before a sample is received. There's a lot of details around service that are important, including customer service, timely answering for questions, and whether the lab will support sending a blood draw, team to the house to get a sample, and then the quality of the lab and the content of the test. Sometimes it was just, I want these genes in this test. This is the only lab that has the gene list I want. The others don't have it, sort of factors. Also policies for following up on variants of uncertain significance, whether they do free testing for family members, and then data return and updates. Do they provide updates and reclassify variants and send out updated reports? And of course, the accuracy of the interpretation. And so this was the final list. I've grayed out the discouraged, not to disparage any labs here, but if you really want to know which ones are at the bottom, I can share that. But we defined 2 preferred laboratories that were broad providers, as you saw in the beginning, a few that were acceptable but preferred for certain areas, and then a list of acceptable, but we will be working to migrate volume away from those laboratories unless there's a specific reason that testing needs to go to them. And this is then defining essentially our roadmap for where we put our resources into integration, like electronic health record integration and other relationships that we can capitalize on and smooth this process more significantly. So that's a lot of what we've been doing in the space of the clinical space and rolling out genomic medicine. I was also asked to talk a bit about how we support research. And so research spans, I sort of bucketed it into population health across our institution, as well as our rare disease efforts, which are managed quite differently. We have a biobank. It has about 120,000 individuals in it. We also, for both phase 3 and 4 of eMERGE, have a major eMERGE program at Mass General Brigham, led by Beth Carlson, Jordan Smoller, Scott Weiss. And then we also have a recruitment site for all of us, as well as at the Broad have a genome center for all of us. So a lot of that is around the generation of data to support population health and lots of health related research, including down here on the right where I've been putting sort of analysis platforms, but that includes the research patient data registry and a lot of the phenotyping extraction from electronic health records to support the research endeavors shown up on the top left. A lot of the sequencing that's done to support both population health and rare disease is largely done at the Broad for most of the high volume projects, partly because the pricing and the volume, we do about between 1,000 and 7,000 whole genome sequencing samples per week. There's actually now, we've launched a public dashboard if you want to watch our volume as it goes up. Of course, we also are supporting 100,000 COVID tests per day, which consumes a lot of the GP platform as well. And then there's some projects that are done at Mass General Brigham in the personalized medicine sequencing core, which is where the LMM clinical lab I used to run was at. Analysis, and on the rare disease side, I'll talk a little bit about our rare disease program. There's actually 2 rare disease programs, one our Center for Mendelian Genomics, and then we also have a site for the UDN program, the Undiagnosed Diseases Network, largely run by Joe Lascausa, Dick Moss, and David Sweetser at MGH. And that's a much smaller volume service in terms of much more in-depth workup of these patients, but on a smaller scale. And then a lot of the support for analysis is on both on Mass General Brigham research infrastructure, computing infrastructure. It's a little more on-prem versus now a lot more use of cloud-based platforms and the Broad's Terra platform. A lot of tools and access to data is being put in that platform. And we've moved our Seeker rare disease analysis tool onto the Terra platform to allow both anyone in our institution, but also anyone around the world can now use it on this platform. We also have a sort of quasi both clinical and research service called the Pathways Consult Service. This was, I think, launched by Katrina Armstrong before I came on in my role, but it really takes MGH inpatients with very unusual and unexplained presentations. Residents spend about 2 weeks on, rotating services to try to really investigate these cases and including connection to research efforts. And that includes doing whole exome sequencing. And my team is, particularly Chrissy, has been doing all of the exome analysis for all of these pathways consult cases. So it's a nice way to bridge our clinical service with the genetic research as well, although reasonably small volume here. So I mentioned our rare disease program, the one that I lead, along with Ann O'Donnell-Luria and Mike Talkowski, started out as a Center for Mendelian Genomics. Actually, Daniel MacArthur and I started it 5 or so years ago. Just recently, that program ended. It was the part of the NHGRI funded Center for Mendelian Genomics. We are now a new center that's part of the Gregor Consortium that Ann, myself, and Mike are the PIs of. And this is our team showed here on the right. And we recruit patients, apply genomic and other omic analyses, focus on methods development, automation, and software for analysis, rapid data sharing, and with the new grant, have some functional investigation as well. And our CMG has really been successful in part because of the amazing set of collaborators we work with. And in the CMG era, when it was funded in the prior era, we're not allowed to recruit our own patients. And so we worked with collaborators who did their own recruitment, and we supported their analysis in a collaborative way. And then they took the candidates and did functional validation and published, were the leads on the publications. And that kind of relationship has been incredibly productive. It's not just been our investigators locally, but investigators around the world. We had some clinical areas of focus, but really would take any orphan rare disease as well. And over the course of that program, studied over 7,700 families with rare disease. We did launch separately, because it wasn't able to be funded by the CMG, a rare genomes project where we could directly recruit patients with rare disease who've never been able to get a diagnosis. And we've been supporting that program. And this is for IRB purposes, we can only recruit from the US, but it's all done online, and we've been able to recruit from all over the US. I think the only state we're missing is either Wyoming or Montana, in Montana. So if you know anyone from Montana, send them my way. But anyway, in orange are all those cases we've returned results. Because we directly recruit, we're able to return those results directly to the patients. But we also identify candidates and need to collaborate and do functional studies. So we have collaborations around the world with other investigators. A lot of our gene discoveries, we build evidence through the matchmaker exchange by finding other cases out there that have a variant in the same candidate gene, and then either bring case level data together to build the story, or case level and functional data together to solve these cases. Largely, we've been focused on exomes as our workhorse. And I think from a cost perspective, that's still our strategy. But we have been, we do still do a fair bit of genome sequencing, particularly when we have tissue from tissue that we can do RNA-seq and combine that with genome analysis. We did a little comparison. This is led by Monica in the team, looking at the added value of genome. And of the 22% of cases from our first 510 we looked at, or that Monica looked at, of the 22% that were solved, 6.7% did require the genome for that solve, versus 18% the answer was actually in the exome, but over time, it required new data, new gene discoveries, or new data on the variant, or new phenotypes, other things that helped actually identify the causal variant within the exome. We also, I didn't mention earlier in the genomic medicine unit, we did do a demonstration project looking at the comparison of genome sequencing to standard of care in a randomized clinical trial environment at Mass General Hospital, working across our different clinics. And we're able to show, and this was just published not too long ago, that a few months ago maybe, that we were able to really detect everything found in standard of care, but then be able to add additional diagnoses by using genome. And this is not, novel, but just being able to prove it to both our own clinicians in our hospital environment, as well as continuing to build evidence for this case. So in summary, from the rare disease work that we've been doing, of all those 7,700 families, we've been able to solve 35% of those. If we just look at novel gene discoveries, that's about 13%. We still find known things that are missed, or these cases just weren't thoroughly analyzed. That's led to 926 novel gene disease discoveries or candidates, about a little over 300 of which are rigorous discoveries, publishable findings, and then another 600 or so are still in that candidate state and need more data. But that does leave 65% of these cases still remaining unsolved, even when we throw everything at them that we have today. And that's, true really of all of the studies that you see out there. In fact, many of you may have seen the recent publication from the UK in their 100,000 genomes pilot on rare disease diagnosis. Their yield, ranged at 25 to 35%, depending on which metric you looked at. And these rates vary by phenotype, what prior testing was done, methods for testing analysis. And there's been many studies published that show the benefit of reanalysis. And we've published some of these studies as well. Just 4 shown here from our the clinical laboratory for molecular medicine, 4% of cases per year getting updates. From MedSeq, 22% of cases when you're looking at their entire genome. In eMERGE, showing the different reasons for updates. And then we just collected the data from the MGH Cancer Center from their clinical service. So not a research study, just routine clinical service. 17% of variants over time since 2015 that they've been tracking all of their variants and their classifications had a knowledge update, 5% having direct impact on patient care. And most of those reclassifications happening in the last 3 years. And why is that? And that's because we're now in an era where all of us are actually sharing data, and knowledge and evidence is just pouring in from every corner. And this is a slide from ClinGen, my one slide on ClinGen, looking at variant classification approaches, disseminating common standards, publicly sharing variant classifications into ClinVar, resolving conflicts between laboratories and engaging experts, all stuff that we focused on in the ClinGen program. And that knowledge that's coming in is really helping inform variants or reclassify variants, things like that. We've also launched more recently a new database called the GenCC, or Gene Curation Coalition, or I call it the ClinVar for gene disease validity claims instead of variants. And so it's not just ClinGen that's making claims on the relationship between genes and disease and how valid those claims are, but many other resources and now private clinical labs are contributing to this as well. We have 12 submitters, over 15,000 Gene Disease Association evaluations to assess the validity. Is this definitive, strong, moderate, limited, disputed, or refuted? About 27% of claims in the literature of a gene disease relationship are viewed as insufficient by experts. So really keeping up to date on this data is critical as we think about it informing the reanalysis of all of the genomes and exomes and panels that we look at. And also, I spent a lot of my time in the Global Alliance for Genomics Health, developing standards and approaches for data sharing across the genomics space. And that all of this is igniting efforts in wide-scale data sharing, but just constantly bringing new knowledge in that we need to constantly utilize and bring into our patient data. So this is my last slide, and then we should have plenty of time for questions and discussion. But the question I get asked all the time is, are you bringing testing back into your hospital? Do you do it in-house? What do you send out? What's your plans for this? We're debating this. Well, so are we. And I can say it's not yet agreed upon. My own view is let the panel testing go to the external labs. In fact, right now, we're letting everything go pretty much outside, but consider ways to bring genomic analysis in-house. And the reason for that is if everything goes out and you're not doing anything inside, you will lose your genetic and genomic professional expertise in your academic medical center. And this space is evolving rapidly. And I don't know exactly when we're going to be there, but every baby born is going to get their genome, and it's going to be, there in the EHR for use over time. And we need to be poised and able to manage that and implement that. And we don't want to lose all of our expertise. And we also, as AMCs, we want to enable genomic data for research. And we get lots of indirects and grants because we have all of our data and we can study our patients. So how do we balance this? And I think where I'm at right now is really thinking about bringing that data back in while it's going out, and really focusing on infrastructure to support reanalysis that may initially be largely funded by research endeavors until there's better reimbursement. But by bringing that data in-house, we can use it for research, and we can start to think about scalable ways to inform reanalysis, which doesn't have to be done on the timeline of a clinically orderable test where the physician wants all sorts of services supported, which you may not be in a position to send blood draws to their houses and do all the things that these clinical labs do. But we can really be innovative in the reanalysis, and that will set us up for the longer term goal where we're really bringing genomics back into day-to-day care of patients, and that genomics is part of our electronic health records and is daily used. And we need to sort of think long term about how to get to that space, but also be practical today that, if we spend all our time bringing clinical testing in-house, we will not be able to spend time on the innovation aspects of this endeavor and position ourselves to be leaders as opposed to followers. So that's sort of where my brain is right now, and thinking about how we think about this in a large, healthcare setting. And with that, I'll just acknowledge the team that I mentioned several times, and some of the projects I spoke about today. But I'm happy to and love to engage in some conversation here.
Heidi, thanks very much. Thanks for stopping sharing. That was fantastic. I will clap on behalf of everyone. It's a weird ending when you end a Zoom talk and then you don't have the audience saying something. But so I will open that up for questions. I will first say that is exactly what my email was about.
Good.
At the end of the that's good. I didn't realize you were going to do that to me. At the end of the webinar, you were talking about infrastructure problems, and we often have high impact talks on science papers and stuff, but ultimately in clinical genomics, it's the infrastructure that's really kind of hindering progress. The science is obviously clearly great, but the infrastructure, and so many of the things you said relate to the problems that we have. As a simple example, we don't know where our testing out goes either, and we have to ask the companies how many tests we've done, and often they'll charge us to give that information back. And that's weird that we've paid them to do the test and we pay them to tell us how many tests we've done. So I will ask one quick question and then I will open it for questions from others. You touched on this in the first half, the infrastructure for the Preventive Genomic Clinic, that one of the challenges is that probably we don't test enough individuals because at the frontline providers don't know how to assess family history. So there isn't a way to capture it in EPIC, and there isn't a way to calculate all these scores. And so there's evidence, documented evidence, that we miss testing on a whole bunch. Clearly in breast cancer, this is my area, I know this for sure, yeah? You said, well, maybe they can come into your clinic, but clearly that's not a scalable option, yeah? A couple of hundred, that's not gonna work.
Yeah.
Do you work with that group to try and improve, because I think improving the flow to the genetics will improve value to the provider and the individuals, yeah?
Yeah, and I think this is where clinical decision support tools are going to be critical, and we are currently evaluating Invitae's GIA chatbot that will walk through not only information about testing, but will actually take the clinical, gather the clinical data from the patient and their family history, and then develop the scores to share that with the physician, so they can sort of walk through. There's points where they can ask for more information and get more details, or they can just say, I already know what I want, click, 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 and sort of get through that upfront counseling piece. It's not, and the ones that say, no, I have more questions, they may still say, I need to see a genetic counselor, but a huge percentage of those could go directly on to testing, probably supported in the primary care environment without a referral out. And then, in the return of results, also with some decision support for negatives to really understand what does a negative mean. We all get concerned that patients overassume what a negative means and the limitations of testing. But I think a lot of that can maybe be even more effectively done with online tools than sometimes an actual visit. And then the patients that we're actually seeing are the exceptions, right? And the positives. Anyone who tests positive after primary care testing does go and get a pretty quick appointment in our cancer clinic. But it's this population that's asymptomatic without, a positive result yet, that are just long in the queue and aren't going to be seen for ages. And I think that's where the clinical support tools are going to be required.
Yeah, fantastic. Okay, I open it up if anyone else has a question. So from Kai, he asked, there are many databases interpreting clinical variants. How do you think the interpretation can be standardized? You obviously are in these efforts, to make sure that happens,
Yeah, so we've, obviously put a lot of effort into this. So releasing the ACMG AMP guidelines for variant interpretation back in 2015, they've been adopted worldwide pretty much. And now there's subjectivity, and we've done some of those studies ourselves to say, even when you've got the same guideline, how you apply the criteria and the codes, does differ. And so through ClinGen, which is now, we now have a second version of the ACMG AMP committee that's been reconvened, and we've been working for the last 2 years actually on the next version. It's now in partnership with ClinGen. And so all of the additional guidance that and more detailed guidance about how and when to apply different rules has been being developed by ClinGen and released periodically on our website, different additional guidance. That'll be brought into this new guideline, and that will be disseminated. And there's, I think so we continue to work on as much detailed guidance as we can. But at the end of the day, there's still an element of subjectivity, no matter how much guidance you give. And that is why the second prong of this is sharing in ClinVar. And we're doing this fairly effectively in the US, but now partly through the Global Alliance and other outreach efforts, really getting every country to share in ClinVar. Because once you both submit and then you see that you've differently interpreted the variant, that tells you interpreted the guidelines differently, and you can start to go in. Or more often, it's one lab had unpublished evidence the other lab didn't have, and that's an opportunity to share that evidence, which we do within ClinGen and we do between labs because they can see each other's submissions to ClinVar. So there's a lot of effort in this space to both standardize approaches, share data to identify when we're still different, apply experts in those cases the labs can't resolve their differences, and really continue to build the evidence base and the accuracy here. But it will be an ongoing challenge.
Yeah. Okay, any other questions? Because if not, I'm going to ask one more.
Well, this is Dan Belisson. I just wanted to say that was an excellent talk, and I felt the pain of an academic lab all through your talk, all the different touch points and all the things we struggle with, that it's actually harder to order a test inside than it is to send it out and have someone do it all for you. And we're constantly barraged with, they do this for free. And I mean, especially in regards to gene panels, where almost every gene panel that's linked to a pharmaceutical type of thing is being done for free. And so it is almost gotta let the panels go.
Yeah.
Thing. So I really like the insight you had on what you're concentrating on. I guess maybe that's the hope for academic labs, is to guard off the genomic space and the data from the genome data, because that is a place, you know, we're getting some traction inside due to the expertise we have, and that kind of testing requires a lot more interaction than a gene panel kind of test, and that's the kind of you know, guidance we can give and is valued. So
that's exactly right. And you know, to really effectively interpret a genome, particularly for a complex indication, you need to have that back and forth with the physician, and it is hard to do that in the commercial reference lab environment. And so there is a lot of rationale for bringing the genomic cases back in, or you know, or even if you do the first round of it, to bring those cases back in for another look. You know, you may, as a clinician, not agree with what was put there as the cause, or feel that you might have reason to be able to look more in depth. And so having access to those genomes, and in fact, building a platform that you're clinician based, and that's one of the things we've been doing with the Seeker platform, is allowing anyone, both researchers and clinicians, to actually look at the exomes or genomes of a case and apply their own. And most physicians aren't going to be in a position to do that, but some of them are. And you know, so I think there's definitely value, and I think that's the areas that we as the AMC should focus on.
Yeah, we've definitely seen the benefit of having a physician geneticist in the room as we interpret stuff, because they immediately look at the patient phenotypes and say they don't have that, you know, that kind of thing. Extremely valuable, and that interchange is really important, I think.
Absolutely.
Yeah.
Okay, any questions? Heidi, can I just quickly follow on from what Dan's question about, because you ended with this very important point on reinterpretation, that's something that AMCs can do, and if you do a genome, it allows for, and how much of the reinterpretation do you mean simple reinterpretation of variants where it changes from a VUS to pathogenic versus reinterpretation of the genome because we have a better way for structural variant calling? How much are you putting in those two kind of bins?
Yeah, it's a great question. You know, my view is we can set up more automated infrastructure, really driving off ClinVar for the most part, to be able to flag that a variant has changed class. And by us doing it at the healthcare system level as opposed to the lab, we're really bringing in all the interpretations in ClinVar as opposed to just the lab's own interpretation, and seeing that where maybe a different lab that's got a newer interpretations come out. So that, you know, is one piece of it, but I'm actually more talking about going back to the primary data files. And that's often the sort of VCF level, the called variants, where you're, you know, rerunning your annotation and filtration strategies and bringing in maybe some updated phenotypic data. Maybe those phenotypic matching algorithms have gotten better, things like that. Occasionally, you might want to go back to your raw, you know, truly raw data and apply new structural variant calling and STR calling and other techniques that have improved over time. But a lot of it is mostly going back to the variant call file and doing that sort of re-analysis of the full data set for that patient.
Yeah, totally. Okay, that makes sense. Okay, any final questions as we head to the top of the hour? If not, Heidi, I'd like to thank you again. That was a fantastic talk. I think it was really exactly spot on the infrastructure challenges, which I think everyone has at the moment. And I think you've really led the way in showing how to kind of tackle them. And I think you've probably given our healthcare system quite a lot to think about. So I thank you again.
Yes, it was a pleasure. And I look forward to further interaction. Take care.
Okay. Bye, everyone.
Bye.